So our next speaker does not need an introduction. This is Dr. Beaudet. Beaudet is going to give us a walk through his life, so I don't think I need to give you a walk through his life. But what I can tell you is that Beaudet took a phone call from me 7 days after Quincy's diagnosis. I'd never met him in my life. He got on the phone on a Sunday morning with me and my husband at 8 a.m., 7 a.m. his time, talked to us for 2 hours. She was 7 days diagnosed and 5 months old, and told us, there's going to be a treatment for this, keep at it. And he was right, and that gave me the hope to go and to help do it and make sure it happened as quickly as possible. So the inspiration he has had on his career, you're going to see, on the impact that has had on all of our lives is pretty tremendous. And so it's amazing to have you here with us. He's going to be speaking to us about what his perspective is from discovery to transformation. Transformative treatments is something that he hoped for, and now he's living to see, which, considering he is the person who discovered the gene, that's pretty amazing. Welcome, Dr. Beaudet. Please.
Okay, well, thank you very much. Allison twisted my arm a bit a few months ago to consider doing this talk, and as I worked on it, I was really glad that I got roped into it. I would say with Allison, you have, I think, a triple threat. She's a mom, she's a scientist, and she's a developer of whatever is needed. And I think a lot of the progress that you heard about, Allison probably has had, if you were to name one person who's had the biggest impact on what's happened over the last decade, I think Allison would be the person. She's provided a spectacular preview of many of the things I'll talk about, although mostly I'll talk about things that happened before your child with Angelman was born. I think I have a couple of things she didn't say that maybe will be interesting. There is a history on the internet. It's got photographs, not much text, but I'm assuming all of you will be able to access these slides by something like a YouTube or something down the road, and that you will don't need to copy down things, but you'll be able to access things easily later. So this is Harry Angelman. He started all of this off. He described 3 children that seemed to be unusually characteristic to him. He called them puppet children. These are 2 of the patients in his publication, and I think you'll agree that they likely had Angelman syndrome. He commented about having seen this painting in Italy, where it's from the 1550s, and there's no speculation that the child depicted had Angelman syndrome, but the child was smiling, and it was holding a drawing of what I think is interpreted to be a puppet. And this painting consolidated his thoughts about the common traits of these children and convinced him to publish. Not much happened in the next 17 years. You can see a case report here and there with 1 or 2 or 3 children. And by 1982, there was a report of 2 brothers. So as Allison has already intimated, there can be familial risks, but the majority of the time it's only 1 child per family. Charlie Williams brought the Angelman story to the United States, I would say, and by 1982 he had 27 cases he could tabulate, and I've zoomed in here on some of the features. The things that were 100% present in those 27 patients were severe intellectual disability, absent speech, and stiff gait. And for all of you who have a deletion child, it's likely this is true for them. From some of the other non-deletion forms, you can have a few words, as Allison intimated. So this was the first scientific development that was relevant, and it wasn't even anything to do about Angelman syndrome. David Ledbetter published in the New England Journal of Medicine that some patients with Prader-Willi syndrome had a deletion of chromosome 15. These arrows point to the non-deleted chromosome, and the deleted chromosome is on the left. And then on the far right, we have a non-deletion case. So not all Prader-Willi patients had deletions, but most did. This is just a closer view of the chromosomes, and I think Allison has mentioned this as well. I talked with Dr. Ledbetter recently about some of the background here, and there was a Prader-Willi meeting in Houston in fall of 1987. This is 22 years after Harry Angelman described the condition. Alan McGinnis, who was a very highly respected cytogeneticist, showed some patients who said, these patients look like they have the same deletion as Prader-Willi, but they don't have Prader-Willi. And Frank Greenberg was a dysmorphologist in the audience, and he said, well, the patients on your slide have Angelman syndrome, and others agreed. So now Angelman syndrome and Prader-Willi are inevitably tied to each other, particularly for the early years. This is Alan McGinnis published a paper shortly after making this point with the deletion. But everybody interpreted it wrong. The chromosome resolution at that time was very poor. It was very hard to get detail resolution of smaller events. But everybody assumed that these were two different deletions in the same region, but in fact there was only one deletion. So my laboratory discovered the first patient with something called uniparental disomy in 1987. We had no idea that it would have anything to do with Angelman. We described a child with cystic fibrosis. But what you can see here in the normal column is there's an egg and a sperm with a pink chromosome 15 from the mother and a blue chromosome 15 from the father. There's a fertilized egg that has one of each, and there's a fetus who has all of this. If you conceive a fertilized egg with a missing chromosome 15, and it's usually the mother who contributes the aberrant chromosome, much like is the case for Down syndrome, and it is an age-related risk. So here you see monosomy. The mother contributes no chromosome 15. The fertilized egg has only one chromosome 15. But occasionally a cell may get two copies of that and be back close to normal, but they will have Angelman syndrome because the two chromosomes are not totally equivalent. The mother's chromosome does something, the father's does not, as you've already heard. If the mother contributes two copies of chromosome 15, you get a fertilized egg with three copies, and if one of those is lost, you can end up with a fetus that has two copies of the maternal chromosome, and this fetus has Prader-Willi syndrome. So this was our understanding up until the point of this publication, where it was discerned that the Angelman deletions were on the maternal chromosome and the Prader-Willi deletions were on the paternal chromosome. And that's depicted here. And now things began to look far more complicated, and there was talk of imprinting, which means that the mother's chromosome and the father's chromosome are doing something different from each other. Then Rob Nichols reported a patient with UPD having Prader-Willi syndrome. So you can see Prader-Willi syndrome is taking the scientific lead on a number of these observations. In this depiction, the black box is a child with Prader-Willi syndrome, and there's 2 copies of the DNA gene that you can see there. The father has 2 copies of an alternative form of the gene, and the mother has the same type of copy that the Prader-Willi patient does. So this father contributed no chromosome 15 to the fetus. This is a picture of some of the people around that time. I mentioned David Ledbetter, Rob Nichols is in the center, and Merlin Butler on the right. So now we had a little bit more understanding that there were 2 forms of Prader-Willi and 2 forms of Angelman. One was the deletion form, and the other was the UPD form. And there were, in this audience, there will be, 1 or 2 families that have a UPD child. So Judy Hall was a very prominent dysmorphologist and geneticist in this era, and she submitted a couple of letters to a journal trying to make sense out of things that other people had published that she thought they interpreted wrongly. The first was a report of an autosomal recessive case of Angelman syndrome, and she looked at the data and said, I don't think this is Angelman syndrome. I think this is genomic imprinting. So this would be a family that had more than one affected child. So historically, the common way to have more than one affected child is autosomal recessive inheritance. But she said this was more likely explained by imprinting, and she was correct. And she also pointed out something that was a little bit remote from what we're talking about, that uniparental disomy probably caused Russell-Silver syndrome. So the synthesis around 1990 was that Angelman and Prader-Willi are the same deletion. The majority of Angelman and Prader-Willi cases are deletion, but non-deletion cases occur. Uniparental disomy occurs in humans. Imprinting explains the differing Prader-Willi and Angelman phenotype associated with the deletion. And maternal UPD, meaning 2 copies of the mother's chromosome of 15, causes Prader-Willi, and paternal UPD causes Angelman. And we were aware that there were more non-deletion cases that were not understood. We had a group of patients we called quad non, that we had 4 things we knew they didn't have, but they still had the condition. So this was, at this point, the scenario. We had the deletions, which were all genetic. We had the UPD, which were epigenetic. If you sequence the DNA of a UPD patient, it will be entirely normal. And it's only DNA changes involving methylation that make the difference. And both of these were de novo. Very, very rare to have a second affected child in a family by these mechanisms. But these are only 2 of the 5 mechanisms I'm going to run through. Then we had something that, again, Allison has introduced you to, that there are 2 sizes of the deletion. Our maps are relatively similar. The larger deletion has some genes on the far left that are deleted. There's some genes on the far right that are deleted with both sizes. Just to give you a little more orientation, here is the UBE3A gene. We'll talk about imprinting centers, which are up here near the SNRPN gene. And you've already heard about the antisense. And so you can see the antisense goes a very long way, but eventually catches up with the UBE3A gene. I've highlighted in blue here the names that I think are associated with these discoveries for the most part. Then a group in Germany recognize differences in DNA methylation. And you can have a restriction enzyme or a reagent that will cut unmethylated DNA but will not cut methylated DNA. And they found a marker that, as you can see in the normal column, there's a larger band and a lower band. The larger is methylated and is coming from the maternal chromosome, and the lower is unmethylated and is coming from the paternal chromosome. And now you can see 3 Prader-Willi patients and 3 Angelman patients tested with this method, and they all show the methylation difference, including UPD. So we had methylation testing was another development in the stage of the diagnostics, that if you did a deletion test, you couldn't rule out UPD, but if you did a methylation test, you could rule out UPD. This is a photo from that era that I obtained actually from unknown sources as to who originally took the picture. But we see Harry Angelman and his wife Audrey. This is around 1990 as far as I know. And Charlie Williams at 2 different ages is on the far right. And Dan Driscoll, who was a player in this work, is also pictured. So again, Allison introduced everything. Right? She said that there are rare families, very rare, and there might be 1 or 2 families in this room who have an inherited form of Angelman. This can be an imprinting deletion, or more commonly, still on? I guess it's back. Okay. So here you can see a family which is staggering how complicated it is. There's all the black symbols are individuals with Angelman syndrome. You can see they're distant cousins of each other. Everybody with a red symbol has the mutation, but they have it on their paternal chromosome, and therefore they are asymptomatic. So any male with the mutation has to first pass it to a female, and then that female has to have a child that is at risk for Angelman syndrome. But that family then is at a 1 in 2 risk for every pregnancy. And I heard about a family in Tunisia when I was visiting there, which had, I think, 5 Angelman children in an impoverished situation. So there are these rare familial parts, and certainly everyone needs to know what form of Angelman syndrome their child has and what the genetic risks are. They're usually going to be very low, but rarely they can be very high. Then in 1994, my laboratory found a gene that we then called E6AP for E6-associated protein. It had to do with a virus interaction. But we found that it mapped in the Angelman region. Now, it turns out that E6AP is UBE3A, its old name and its new name. We looked for whether it was imprinted or not, and we looked in fibroblasts and lymphoblasts, and on the left you can see data for SNRPN, which is the gene expression in the paternal chromosome, and you can see for the 3 Prader-Willi patients, it's not making any product. But for Angelman syndrome, this gene was turned on in fibroblasts and lymphoblasts, and so we thought, well, it's not imprinted, so it must not be the Angelman gene. We were totally wrong. I'll come back to that. This is a family, again, that we studied that had one of the first imprinting center deletions. It's a family with Prader-Willi. The deletion is causing an abnormal pattern of DNA that wouldn't be in a normal father or a normal mother, and that band is present in the father and the 2 affected children with Prader-Willi. And you can see the methylation pattern we've seen before. So we knew we could map precisely where this deletion was. And here we see mapping of 3 deletions of the imprinting center causing Prader-Willi, and 2 imprinting center deletions causing Angelman syndrome. And the critical location is a little bit to the left in this slide for the Angelman imprinting center, and a little bit to the right for the Prader-Willi imprinting center. So now we have yet another group. We have the deletion, we have the uniparental disomy. Now we have imprinting center mutations, some of which are de novo and some of which are inherited and occasionally can give rise to the giant type of pedigree that I showed you. But imprinting center defects are not all the same. Some are deletion, some are not deletion. So then, 3 years after we had figured out where the UBE3A gene was, Joe Wagstaff and a trainee in his laboratory reported mutations in UBE3A that caused Angelman syndrome. What you can see here in the top is a sequence for the normal gene, and below a sequence for an individual who has both the normal and the abnormal gene. The abnormality is a 5 base pair insertion, so the gene is disrupted functionally by this insertion. Our laboratory at the same time found the same thing, that there were mutations causing Angelman syndrome. Here there's a mother with a normal pattern, and then the lower plot is, again, 2 DNA sequences running together, and now you see on the right-hand side there's 2 peaks at a time instead of 1 peak at a time. And then it turned out it was easy to find lots of mutations. There must be hundreds of mutation families now identified. This was 20 or so identified back in 1999. And there are a couple of reviews out there for anybody who might be interested of things up to this point. They're indicated here, and they are reviews about Prader-Willi and Angelman, because everybody was thinking these somehow go together, and they go together for scientific meetings. Sometimes they go together even for family support group meetings. And you see the Prader-Willi patient with obesity on the left and the typical Angelman patient on the right. I'm sorry, other way around. And so now we had multiple forms of all of these conditions. We had deletion Prader-Willi and deletion Angelman. We had UPD Prader-Willi and UPD Angelman. We had mutations in about 20% of patients for Angelman, but no mutations have been identified for Prader-Willi that cause Prader-Willi syndrome, probably because you have to damage multiple genes to get the phenotype. And then there are imprinting mutations, as I've mentioned. So now we got 4 out of our 5 categories, this one being now point mutations, and these can be de novo or inherited. These can give rise to multiple affected siblings and to gigantic pedigrees like you saw. We were able to get some mice with UPD from a famous geneticist in the UK. And so what you see here is a mouse with paternal UPD, that is 2 paternal copies of the gene, and a mouse brain from a normal. And the red signal is detecting the Angelman gene. You can see it is on in the normal, but there's no signal in the paternal UPD. So this was clear evidence that this gene was imprinted in brain, and it took us a while to put the whole story together. At this time also, the Human Genome Project was underway, and now you see a very large map of this 5 to 6 million base pairs that Allison addressed across the region. And then each line, each horizontal line, is what we call a YAC, or yeast artificial chromosome, that had a particular chunk of the genome. So you're looking here at 5 or 6 million base pairs, and I've circled the location of the SNRPN gene, which is near where the imprinting centers are, and the UBE3A gene. So the Human Genome Project was formulated in the 80s and planned. And it was officially launched with federal funding in 1990. They declared a quasi-complete genome in 2003 that had about 92% of the genome. And then you can see is quite a long time later in 2021, they had what they then considered a more complete genome with only 0.3% of the DNA still not understood. This genome project made everything in genetic 1,000 times easier. And everything you want to do on Angelman became 1,000 times easier for the genetic manipulation. Then came the antisense. It only came along in 1998 when a group described, you can see running down the middle from left to right, a double-stranded DNA molecule. And on the top, the RNA is being copied in one direction in red and in the other direction from the other strand of the DNA. And this represents the sense strand of UBE3A and the antisense strand. Then came Angelman mice. The first came from Youngwei Zhang, who's, I think, here and talking in a few hours or minutes. And so he made a copy of the mouse gene with damage to exon 5. And this was then introduced from embryonic stem cells into the mouse germline. And if you bred the mice on the left with the mutation on the maternal chromosome, you get an Angelman mouse. And if you breed it with the mutation on the paternal chromosome on the right, you get a healthy mouse. And the Angelman mice had a lot of features of Angelman syndrome, and they were considered to be, while they don't talk and have many intellectual functions, that a lot of their abnormalities seem to parallel what we knew about Angelman syndrome. This is just a depiction of an Angelman mouse brain. With the left is the both maternal and paternal copies of the gene are normal. In the center, there's the normal maternal copy that is turned on and giving red signal again. But in the one where the maternal copy is damaged and the paternal copy is normal, you see no expression. Then came a memorable meeting in Finland in the year 2000. I learned preparing this talk, and this is the sauna capital of the world, Tampere. I assure you that everybody has a bathing suit on in the picture. It was a very different environment. I had never been to a location where it was the longest day of the year and it was light all night long and everything. But it was a really key point in my thinking and maybe our thinking about Angelman syndrome. This is the brochure from that meeting, thanks to Terry Jo Bichelle, who found this for me. And we had begun to think, I had been interacting with Huda Zogby, who worked on Rett syndrome, and they were thinking about DNA methylation and turning on genes. And we began to think about the possibility of turning on the father's copy of the gene. And this is Terry Jo in 2000 with her son breastfeeding in the lobby of the hotel at 3 a.m. And I was down there with a laptop. I do think I had a laptop by 2000. And Terry Jo comments that I firmly believe that even then that Angelman syndrome could be cured by activating the dormant paternal gene. And I started saying something that Allison and all of you would welcome, that I thought there was a remarkably favorable situation for trying to treat Angelman syndrome it was much more favorable than hundreds of other genes and conditions out there because we had this option of potentially turning on the father's copy of the gene. So at that time, we started 2 trials with different folic acid regimens trying to increase methylation on the father's copy of the gene. And 10 years later, these 2 publications appeared and demonstrated that you could not turn on the copy of the gene from the father using folic acid. Along came another afternote, kind of, that I don't know if there will be any families in the room who this applies to. There was a report that in vitro fertilization can cause first another syndrome, Beckwith-Wiedemann syndrome, but then a few patients with Angelman syndrome. And there are 3 patients here with Angelman syndrome. ART stands for artificial reproduction technology. And ICSI is intracytoplasmic sperm injection. So at the cell in the culture dish, they take a copy of a sperm up in a pipette, they punch it into the egg, and they squirt it out. So this is, again, maybe one of the rarest forms of Angelman syndrome. It's not quantitatively a significant risk for people using reproductive technology. But it can be tested if one has IVF. You can test if you might have this Beckwith-Wiedemann or Angelman syndrome in the fetus. So now we have the full story. De novo mutations, de novo UPD, imprinting defects. And now I've separated out this category of imprinting defects that are not associated with any change in the DNA sequence, but only with the DNA methylation, and then the UBE3A mutation. So my laboratory then generated another mouse, and this is a mouse with brain cells turned green. And so Scott Dindo, who's had a role in a number of the studies that we've been talking about, created a mouse where if the green tag is on the end of the Angelman protein, so if the Angelman protein is expressed, you see the green tag. If it's not, you don't. So you see on the left, when you have the reporter gene on the maternal chromosome, the cells in the brain are green. When it's on the paternal chromosome, the cells in the brain are not green. But you can do a drug screen to try to find drugs that will turn on the father's copy of the gene. This just shows that the distinction was strong and good for screening because it was a kind of an all or none. You see the maternal brain, the paternal brain without the yellow-green blobs, and then just zooming in on the right. So Ben Philpott then reported what I think probably is the most important paper that really changed thinking in 2011. So he took this mouse that we had made, and these mice are available to anybody. We weren't aware what he was doing. But he introduced the term unsilence and screened for drugs that would turn on the father's copy of the gene. So he took the brains of the mice, put the neurons or cells in cell culture. He exposed the cell culture to a drug screen with thousands of drugs, and he found that a group of drugs described as topoisomerase inhibitors activated the paternal copy of the gene. So this was very exciting. There were some who were even saying, well, maybe we should try it in patients right away. But these topoisomerase inhibitors are quite toxic. They're used in cancer chemotherapy. And so no clinical trials using those were carried out. This is just one picture from the publication. If you just look at the one panel in the center, there are 2 green lanes, which are the reporter gene on the maternal chromosome. There's two center lanes where it's the reported gene on the paternal chromosome and it's turned off, but when you add topoisomerase in the right two lanes, you see that there's this bright green signal. So I think this became the mantra. I think that turning on the father's copy of the gene has many advantages potentially. We believe it's regulated exactly like the maternal copy. And I think that it's controlled and turned on in the right cells in the right ways, and it's not turned on in places where it shouldn't be turned on, and we don't think it would be overexpressed. So it's a really, while we talk about gene therapy for everything, and I think gene therapy has been and will continue to be explored here, it's rare that you can get gene therapy to go in there and go into just the right cells, do just the right things at just the right times, and even make multiple different forms of the protein called isoforms. So I think that gene replacement therapy has potential disadvantages compared to activating the paternal copy of the gene, but clinical trials may say, no, the patients do better with gene therapy, so that's the answer. Lin-Yan Meng in my lab created a mouse with a DNA or RNA transcription stop signal to stop the antisense, and this just shows the antisense and the sense are really colliding right in the middle of the Angelman gene. So the first part of the Angelman gene is made, but doesn't allow it to make a whole protein. The antisense and sense collide, and in the lower panel there you can see, again with the brown, that in the Angelman you have no brown, but if you introduce the stop signal, you can bring on the brown color. Then there was this publication where we initiated a collaboration with ISIS, now Ionis. You can imagine why they changed their name. Here we showed that ASOs or oligonucleotides could activate the paternal copy of the gene. I won't take you through the panels, but that was the bottom line. And this, I think, everybody did think immediately, how quickly can we get this into the clinic? But this publication was in 2015. We're already 9 years down the road from this publication, and we're still in the early stages of clinical trials. So this is my view of where the molecular genetics are at the moment. A lot of it fits with what Allison said, although I would maybe have one different emphasis. You can have gene replacement therapy. I think it would be better to use gene therapy to turn on the father's copy of the gene for reasons I've already stated. In terms of the father's copy of the gene being turned on, you can have ASOs, you can have gene therapy, you can have small molecules, you can have CRISPR, and there's interestingly some progress in delivering gene therapy vectors into the vein that will then go to the brain quite efficiently. So in red, I've got what I think is the intermediate to long-term desire to have a once-per-lifetime injection or have a pill, which doesn't have as much difficulty in administration. The other is at the top there to upregulate the other deleted genes, as again Allison pointed out. I would say this deserves a pillar of its own. If you have a deletion child, which is true for 70% of the patients, you can hope for an 85% improvement, which would be a gigantic improvement. I would think they would be talking and communicating very well. But if you want 100% correction of the deletion patient, you have to deal with these other genes. Allison showed you that some are essential, others are not essential. There's one curious thing, that one of the genes that's in this category is an albinism gene. And you may be aware that deletion children are usually somewhat lighter complexion than their siblings. And this is because there's an albinism gene and there's only one copy of the albinism gene. But it's also one that we say, well, but you can live with a child with lighter complexion. We don't need to fix the albinism gene. So you can try to get a full understanding of all of these genes and know which ones you need to fix and which ones you don't. But I think this needs some kind of sub-pillar in Allison's story, and I think that what we really, I think, want to come back and find that there are 5 different groups working on the other deleted genes instead of one. You'll hear a talk from Dr. Atuv, I think remotely in a few minutes, who's focused on this approach, which I think is very important. And so, what are the biggest positives? One is the option to unsilence the paternal copy of the gene, the potential for a lifetime, once per lifetime activation. And there's one thing that Allison didn't cover is just recently, in July, there's a publication from Ben Filmhout's lab doing additional screening, demonstrating molecules that might function as a pill to activate the paternal copy of the gene. Those of you who have interest in writing down a number, the PMID, if you want to go on PubMed and look at this paper, it's 389-77672. So I think prominent consideration of a pill rather than a non-pill strategy is important. And 2 of the talks today, I think I mentioned, are particularly relevant. One is by Dr. Zhang about using CRISPR to directly activate the paternal copy without gene therapy. And then the other I've listed there is Dr. Atuv, who's going to talk about how to deal with the other genes. So I'll stop at that point. Thank you. I had one other thing that I wanted to say. I didn't try to tell the story of what Angelman Syndrome Foundation contributed for decades and existed and was, very active at the time of the Finland meeting, and then what FAST has contributed. But somebody could do a presentation like this about what the role of ASF has been and what the role of FAST has been, and I think it would be interesting history to have access to.